What if you were born with only 23 chromosomes? Not 23 pairs, just 23 chromosomes in every cell of your body. At first, that might sound like a smaller version of a normal human, but it would not be, because those 23 chromosomes are not simply half of you. They would mean that almost every cell in your body carries only one copy of each chromosome instead of two, and that raises a much stranger question. Could a human body actually develop that way? To understand the answer, you first need to fix one very common misunderstanding. You have probably heard that humans have 23 chromosomes. That is true, but only for certain cells. Your ordinary body cells usually contain 46 chromosomes, arranged into 23 pairs. One chromosome in each pair came from your mother. The other came from your father. Your egg and sperm cells are different. They contain only 23 chromosomes. This is called being haploid. When an egg carrying 23 chromosomes meets a sperm carrying 23 chromosomes, the two sets combine. 23 plus 23 gives you 46. That first cell, the zygote, then starts dividing again and again to build a human body. So the 23 chromosomes in an egg or sperm are not an incomplete human genome. They are one complete set designed to be paired with another set. And this distinction is the key to the entire thought experiment. In fact, in 1956, Tjio and Albert Levan published their famous paper, The Chromosome Number of Man, establishing that humans normally have 46 chromosomes rather than the 48 that had previously been accepted. That discovery changed human genetics, because once scientists knew the normal number, they could start asking a much more important question. What happens when the number goes wrong? Now imagine that instead of your cells having 46 chromosomes, every cell in your developing body had only 23. Not because one chromosome was accidentally lost, not because of a small mutation, but because your entire body was built from a single chromosome set. You would be haploid. And here is where things become fascinating. A haploid human has never been established as a naturally viable human condition, so nobody can simply tell you exactly what such a person would look like. This is a hypothetical scenario. But biology gives us some very strong clues about what would probably go wrong. The first problem is something called gene dosage. Think of your chromosomes as instruction libraries. You normally have 2 copies of most of those libraries. That does not mean every gene is switched on twice as strongly. It means the cell has 2 copies of the genetic information and can regulate their activity in a very controlled way. Remove one complete set, and suddenly the balance of thousands of genes changes. Some genes can tolerate having only one working copy. Others cannot, and development is especially sensitive to these dosage changes. Your body is not built by simply reading genes one at a time. It is built through enormous networks of genes switching other genes on and off, controlling when cells divide, when they specialize, where they move, and what tissues they become. Change the dosage across the entire genome, and you are changing the instructions for the entire construction project. But there is an even deeper problem. Your chromosomes do not just carry DNA sequences. Some genes also carry what scientists call genomic imprints. In simple language, the cell can chemically mark certain genes depending on whether they came from the mother or the father. That means 2 copies of a gene are not always biologically interchangeable. The maternal and paternal genomes can carry different developmental instructions. Some of the strongest evidence for this came from classic mouse experiments in the 1980s. M.A.H. Surani, Sheila Barton, and Michael Norris studied embryos containing unusual combinations of maternal and paternal genomes. They found that embryos containing only maternal or only paternal genetic contributions could not complete normal development. Around the same time, James McGrath and Davor Solter independently showed that mouse embryos needed contributions from both parental genomes for normal embryonic development. These experiments helped establish the concept of genomic imprinting, and this matters to your 23-chromosome thought experiment. A human with only one chromosome set would not simply have less DNA. The normal developmental relationship between maternal and paternal genetic information would also be fundamentally different. There is another clue from a real human condition. It is called Turner syndrome. In the most common form, a person has 45 chromosomes instead of 46 because one X chromosome is missing. That is called 45X. Their work pointed toward major differences in gene expression, including in tissues involved in placental development. So if losing just one chromosome can create such a severe developmental problem, what would happen if you removed an entire second set? The answer is probably much more dramatic. A true haploid human embryo would have only one copy of chromosome 1, one copy of chromosome 2, one copy of chromosome 3, and so on. That means the dosage of thousands of genes would be fundamentally different from normal human development. And the problem would begin very early. Remember, after fertilization, the zygote has to divide. One cell becomes 2, 2 become 4, 4 become 8. And eventually, these cells begin taking on different identities. Some become nervous tissue, some become muscle, some become blood, some become placenta. Every one of those decisions depends on carefully controlled gene activity. If the entire genetic dosage is wrong from the beginning, the developmental program may simply fail. But there is an even stranger question. Could a 23-chromosome human cell survive at all? A single human haploid cell can absolutely exist. Your sperm and egg cells are examples. So the problem is not that a cell with 23 chromosomes is automatically impossible. The problem is building an entire complex human organism from cells that remain haploid. That is a completely different challenge, and this distinction is important. Your body already contains cells with unusual chromosome numbers. Some cells can become polyploid, meaning they contain extra sets of chromosomes. Some cancers contain abnormal chromosome numbers. And during normal cell division, chromosome numbers can temporarily change as chromosomes are copied and separated. So biology is not obsessed with the number 46 in every single cell at every moment. What matters is whether the chromosome number and genetic dosage are compatible with the cell's job and the organism's development. Now, imagine this hypothetical person somehow survives the earliest developmental barriers. What would happen later? This is where science becomes much less certain. There is no naturally occurring, healthy adult human with a complete haploid genome that scientists can study as a reference. So it would be irresponsible to say, you would definitely be shorter, or you would definitely have a particular face, or your brain would definitely work in a particular way. Those are guesses. What can be said with confidence is that normal human development would be profoundly disrupted. Growth would likely be affected. Organ formation would likely be affected. Placental development would likely be affected. Cell division and tissue specialization could be severely disturbed. And reproduction would raise another enormous problem. If you were a haploid human and somehow produced ordinary haploid sperm, or eggs, combining two haploid cells would give you only 46 chromosomes in the next generation. That part sounds normal. But the biological machinery required to produce gametes is built around a diploid life cycle. Humans evolved a very specific system: diploid body, haploid gametes, then diploid embryo. A permanently haploid human would be outside that normal reproductive system. And there is something even more fundamental. Evolution has shaped humans around 2 copies of our chromosomes. That means the human genome is not simply a list of genes, it is a system. Gene dosage, chromosome pairing, genomic imprinting, DNA replication, cell division, development. All of these processes are connected. Change the chromosome architecture, and you are not changing one feature. You are changing the operating system underneath the entire organism. So would a human with only 23 chromosomes be possible? Based on what we know today, a naturally viable human with a complete haploid set in essentially all body cells is not an established biological possibility. But the thought experiment teaches you something much more interesting. You do not have 46 chromosomes because biology randomly chose a number. You have 46 because your entire developmental system has evolved around 2 coordinated sets of genetic information. And those 2 sets are not simply duplicate copies. And that is where the thought experiment becomes a lesson in what makes human life possible. One came from your mother. One came from your father. They interact, they regulate each other. They help build you. So the next time someone says, humans have 23 chromosomes, remember, that sentence is both correct and completely misleading. Your sperm or egg may carry 23, but you, the person watching this, are built from 23 pairs. And that tiny difference between 23 and 23 pairs is the difference between a single genetic set and the extraordinarily complicated biological system that became you.